name is?
Jeff.
And your age?
He's 51.
And what were some of the notable events during this past year?
Well, we took a road trip.
That's right, I drove on I-95.
And where were you going?
Maine.
Actually, Massachusetts.
Yeah, Massachusetts.
Jeff, once an avid golf player, could no longer keep score. He could not draw a simple outline of the United States, nor remember major cities. His wife Sarah brought him to Dr. Daniel Pollen, hoping he could be diagnosed and treated. She then presented the doctor with a family pedigree that would change Pollen's life. The pedigree showed 4 generations with over 100 family members. It also showed the relatives affected by Alzheimer's disease, a slowly progressive degenerative brain disease that first impairs and eventually destroys memory and all higher processes. 1/5 of the family had experienced these terrible symptoms. And it all began with Hanah. Hanah was born in Babruysk, Russia in 1844. In the middle of her adult life, Hanah began to struggle with her recent memory and personal care.
I was Hannah's 6th child, and when she became more ill, it was my job to assist her. I had to comb her hair, I had to help her remember basic information. She remembered the events from her youth, but she could not recall the events of the day before.
In 1906, Alois Alzheimer and colleagues Franz Nissel and Emil Kraepelin discovered bundles of fibers seen in microscopic examination of the brain over the entire cortex of people with dementia. There were also multiple foci called plaques present in people with presenile dementia. It would later be found that these plaques were made of the protein beta amyloid. The symptoms of this disease, which would later be named after Alzheimer, included amnesia, the impairment of memory, apraxia, sensory abnormality, and agnosia, the inability to recognize familiar objects. During the early 1900s, 8 of 9 of Hanah's children immigrated to the United States. Some of the older family members began to display symptoms akin to Hanah's, and more descendants passed away, young and senile. In the 1940s, Charles, a great-grandson of Hanah, began research on the familiar Alzheimer's disease that plagued his family. He found that the mean age of the onset of the disease was 46 to 47. By this time, 4 out of 8 of the American members of the second generation had developed the disease. It was noted that Alzheimer's had been passed mother to daughter, mother to son, and also father to son, meaning it was not a sex-linked disease. In his investigations, Charles began to wonder if there was a linkage between familial Alzheimer's disease and blood group antigens. He collected blood samples from 75 members of his own family, assigning the status of unaffected, escapee, at-risk, or unaffected spouse to each. Bringing his project to the attention of expert Timur Greenwald, the two soon concluded that Alzheimer's disease in Charles' family was not linked to any blood group. After the study with Greenwald, Charles decided that the best available research option would be the study of RNA or protein synthesis in Alzheimer's disease. Robert Terry and Saul Corey took up the project, using recent advances in electron micrography. They studied the gray matter and white matter of the affected brain, but concluded all chemical differences were secondary to the known loss of brain cells. Thus, the scientists could not yet distinguish the cause from the effect. In 1979, with the Alzheimer's disease becoming more and more publicized, the Alzheimer's Disease and Related Disorders Association, or the ADRDA, was founded. Its purpose? To study, promote the knowledge of, and eventually cure Alzheimer's. The disease, said Roberta Custard, that is the funeral that never ends. In 1983, Peter Hislop was chosen to be the principal investigator for the Alzheimer's Disease Project. In 1958, it had been found that Down syndrome was caused by an extra copy of chromosome 21. In the mid-1970s, there had been a link found between Down syndrome and Alzheimer's disease. Now, Hislop concluded that the genes for Down syndrome and Alzheimer's might be on the same chromosome. He noted that victims of Down syndrome develop Alzheimer's with an unusually high frequency by middle age. Furthermore, relatives of patients with Downs have a small but statistically increased chance of developing Alzheimer's. Dr. Hislop's ideas
along with Jeff and Sarah's visit to his office, encouraged Dr. Daniel A. Pollen to investigate the terrible Alzheimer's disease. Pollen used an Italian family, a German family, a British-Canadian family, and Hanah's family in his studies to determine if Alzheimer's was located on chromosome 21. Pollen used load scores, statistical tests which compare the likelihood of two loci actually being linked to the likelihood of random coincidental data in his experiment. His paper, Genetic Disease Causing Familiar Alzheimer's Disease Maps on Chromosome 21, was published in 1987. This paper concluded that there was a high chance of the Alzheimer's gene being on chromosome 21, particularly in the Italian family. Even more interestingly, the gene that produces amyloid, a polypeptide aggregate with various structural configurations, had also been found to be located on chromosome 21. Could the genetic defect in Alzheimer's disease in fact be an abnormality in the gene that produces the amyloid protein? If beta amyloid, frequently seen in afflicted patients, was unique to Alzheimer's, then it could have been a possible test for the disease. However, the Alzheimer's gene and amyloid gene were close, but not close enough to be definitely linked. There were still approximately 8 to 10 million base pairs between the enhancers of the two. Later in 1987, Lucy, a 51-year-old great-granddaughter of Hanah, self-diagnosed herself with Alzheimer's and submitted herself to testing. Her load score linked two DNA markers towards the centromeric, or middle, part of chromosome 21. Thus, Lucy set a limit on how far the abnormal gene must be from the centromere, and how much of the chromosome towards the telomere ends could be excluded from further analysis. Though they had determined that the Italian family in particular had strong ties with chromosome 21 as the cause of their familiar Alzheimer's disease, Hanah's family and others did not display a strong link. Thus, the scientists postulated that the symptoms were a result of an abnormal gene on chromosome 21, but that the cause and activation portion of Alzheimer's disease was located on a separate chromosome in certain familial cases. In 1991, Dr. Gerard Schellenberg decided to construct a linkage map for chromosome 14. He later said that the chromosome had been neglected because there were very few useful DNA markers on it. However, after his intensive studies, he noted that a German family with familial Alzheimer's was significantly linked to chromosome 14, with a peak load score of 4.89. Later, using Hanah's family's information, a peak load score of 2.17 was found. Thus, Schellenberg concluded that there must exist another chromosome locus that affects Alzheimer's in certain familial cases. He had established the evidence for linking familial Alzheimer's disease to different causer genes for each family. In Hanah's family's case, this causer gene was located on chromosome 14. After nearly a century, the cause of Hanah's disease had finally been discovered, but the question of the cure still remains.
The disease, for all its horrors and ironies, has brought our family many gifts as well. Throughout my childhood and into young adulthood, I was taught to live life with a sense of responsibility, but with humor and compassion. Having had to face the possibility of joining my mother and my uncles and aunt on that horrific mountaintop called the family disease, I have learned the value of being able to function and contribute. I long for the day that my niece and nephews will be released from the ghosts of their ancestors.